Good afternoon. I would like to welcome everybody for this afternoon section. I would like to introduce Dr. Angelica Cibrian from National Genomics Laboratory for Biodiversity of CINVESTAV in Mexico, and thank her for accepting our invitation to present at BSB. Angelica Cibrian holds a PhD in ecology and evolutionary biology from Columbia University, and she's affiliated to New York Botanical Gardens Genomic Lab of the American Museum of Natural History, and also a member of the New York Plant Genomics Consortium, investigating the genome evolution of seed plants. Her research focuses mainly on the conservation genetic of economically important plants and the phylogeography of their insect pests. Today, she will talk about the evolutionary genomics of ancient plants and their symbionts, from species to metabolites. Thank you, Dr. Jamarillo.
Thank you for the introduction and the kind invitation to be part of this symposium. I'm very happy to be here. I'm going to proceed to share my screen. I am currently based at Langevio in Mexico, in CINVESTAV, where I have been a professor for the last almost 10 years. And part of the work that I will share today is the result of that effort in Mexico. All right. So the title of my talk is Evolutionary Genomics of Ancient Plants and Their Symbionts, from species to metabolites. And this is a result of the joint collaborative effort of two groups of research, Paco Barona's group, also at Langevio, and myself, as well as a couple of lab technicians. These are some of the students that have participated in this effort thus far. So cycads are gymnosperms. They're plants that do not have flowers. They're from the order of Cycadales. They are of subtropical distribution. There are not many species, 250 species, but they're remarkable in many ways, including the fact that they are very long-lived. They have even lifespans that can go as high as 2,000 years old in that have been around now for 300 million years. And they pretty much have endured and survived exchanges of pollinators, herbivores, predators, climatic changes, soil changes, atmospheric changes, and essentially are survivors of a life that was that has many life forms that have gone extinct before. So cycads are also, unfortunately, at the moment, the most threatened groups of plants on the planet. They're Cycas appendix 1, and unfortunately, they're disappearing, even though they have survived for millions of years. They're now disappearing due to habitat loss and deforestation, mostly deforestation, but also to illegal traffic. However, when they are found, they're again remarkable because they can thrive in soils with very low nutrients, either in content or in access. And they usually live where no other plants can live, especially where angiosperms cannot tolerate very difficult soils. And so this makes them a very interesting group to explore, given their extreme adaptation to these very poor environments in terms of soil nutrients. They are also incredibly tolerant to fire. So they're not completely fire resistant, but they are quite fire tolerable. tolerant. So they are also very interested in that sense. They retained very ancient features, and so they are also known as living fossils. And I'm not going to go into the anatomical details, but they essentially have morphological features that are more similar to ferns and ginkgos. They have exposed seeds and large cones that you may at some point see in botanical gardens, or perhaps in some of the cycads that are used as ornamentals. But more fascinating for our group is the fact that they maintain very long or very ancient relationships with insects. They have at least 100 million year old pollination mutualism with beetles, in which they have developed a brood site pollination system in which beta-myrcene mediated pollination takes place. And this is a long time before angiosperms even appeared on the planet, and cycads were using these compounds to attract and repel insects as part of their pollination mechanisms. Not only weevils, but also thrips, which are distributed worldwide in different genera of cycads. They also maintained a very ancient relationship with aposematic herbivores, and these are butterflies whose caterpillars eat their fresh leaves and retain toxins that are unique to cycads, and this is something that I will mention a little bit in a moment in more detail. So what you see here is the larvae feeding on fresh dying leaves and some of the butterflies that we have reared in the lab. Cycad chemistry is unique among plants. They are highly toxic plants. They have actually glycosides, cytosines, glycosides, and a non-proteinic amino acid that is called BMAA. And this level of toxicity has been linked to human intoxication and has led to the hypothesis known as the cycad hypothesis of human intoxication. And this is because there was a high incidence of ALS-PDC, which is a neurodegenerative disease in humans, in the Pacific, some Pacific islands in last century. And so it was proposed that BMAA that was contained in high content in the seed of cycads was actually transferred to humans because humans like to consume cycad seeds in the form of flour in other ways. And also they were consuming bats, which are some of the animals that eat cycad seeds. So this BMAA metabolite has been a fascination both in the literature, as you see here in the Island of the Colorblind of Oliver Sacks, but also in the medical community because of this potential link. However, our group has done some research showing that that's actually not necessarily the case, but that is not to say that cycads are not toxic. Cycads are extremely toxic to mammals. Cycads also have a coralloid root, and this is a specialized organ, a specialized root that looks like a coral, and it's apogeotropic, and this means it grows above ground. And its basic function, its basic known historical function, is to fix atmospheric nitrogen, mostly through cyanobacteria that are inside this root. However, our work has also showed, and this is part of what I will be showing today, that this coralloid root can also produce metabolites. And there are other bacteria, not only cyanobacteria, fungi, and viruses. This is a cross-section of a coralloid root. And what you see here is the cyanobacterial zone in green, which actually has been studied. And these are like the close-up of bacteria that are hosted inside the coralloid root. So part of our hypothesis is that cycads can survive in nutrient-poor habitats and can maintain specialized biological associations by harboring endosymbionts like cyanobacteria and acquiring their toxic metabolites via the coralloid root. And this has allowed them to have this adaptability to extreme environments and has allowed them over time to survive as a lineage. We also have a second hypothesis that these endosymbionts, at least some of them, mostly bacterial symbionts, are transferred to insects that are those pollinators and herbivores, and therefore are maintained in this whole cycad biological system. In this transfer, bacteria would go from the soil into the coralloid root and pass through the plant cones, where beetles would feed, get stored as well in the leaves, where butterfly larvae would eat, and then would come back to the soil in a cycle that actually ensures that functional bacterial symbionts are maintained. So these two hypotheses, we've been testing these two hypotheses now for a few years, 8 years almost, in which we first began with extensive fieldwork to find both correlate roots and SICA-diverse insects, including family members that do hard work. And we then do a combination of greenhouse work, microscopy, wet lab, and big data, mining genomes, metagenomes, transcriptomes, and using several pipelines of bioinformatics that I will mention in a moment in more detail. In the wet lab, we have had to do several innovations with respect to these difficult samples because correlate roots are actually quite difficult to get. They're within rocks or very basically it's fragile when you sample them and they can disintegrate. So what we've done is that we have done a combination of methods in the wet lab to be able to harness as many bacteria as possible from the correlate root. And this is a traditional amplicon sequencing from 16S Illumina iText. We have also done traditional strain isolation from where we do whole genome sequencing. And we have done what we call the community co-cultures, in which we enrich for one particular taxon, and then we allow others to grow with it. In that way, we actually enrich the community that was sampled originally from the either insect or root. And from there, we can obtain very deep metagenomic sequences because these taxa are enriched in this community culture. And from there, when we find key players, we actually can pull them apart and play with them or test different interactions between them in semi-synthetic communities, for example, cyanobacteria and colobacter. And these different strategies give us a glimpse of both community interactions, but also taxonomic resolution, depending on where you are in this different methods. And so this is what a co-culture looks like. This is one of our early co-cultures. And even though it looks more like just a little piece of, just a little bit of dirty water, actually it's a bacterial community that retains microbial interactions and in which you can test very carefully and quite deeply public goods microbial community ecology that is taking place there. These have been alive now for more than 4 years. And you can use a bunch of different techniques that you can apply to this co-culture to measure different traits of the interactions. So in terms of the bioinformatics, to be able to characterize these communities, we have been focused on thinking about biosynthetic gene clusters. So once we have the original, once we have the environmental sample and we have either co-cultured it or sampled it directly, what we want to do is, on one hand, we want to understand the taxonomic composition. But on the other hand, and this is more important to us, we want to understand the function, because we know that many bacteria are going to be shared, but we don't know what they are doing. And so what we have done is we have focused on identifying and understanding the evolution of biosynthetic gene clusters. And these are just linked genes, physically linked genes that participate together in a specialized metabolic pathway, usually regulated by one or two regulatory elements. And so you can imagine here you have a canonical BGC, biosynthetic gene cluster, for a particular pathway, bacterial metabolic pathway, in which you have a green gene, a blue gene, and a red gene. And these BGCs can change over time and in different populations in various ways. They can expand with duplicates, they can recruit new genes and have a slightly different function, or they can be minimized depending on their use or adaptability to the particular genomes. And these, once you know the sequence from these biosynthetic gene clusters, you can actually do predictions of the natural products or specialized metabolites that these BGCs are producing. So for us, identifying these BGCs is key. And so to do that, we have, and this is a very close collaboration with the Barona lab at L'Ancreville, we have used a computational framework for a systematic genome mining of biosynthetic diversity. And so what does this mean? Essentially, we take advantage of very large databases that are already in place for bacteria, such as anti-SMASH, which is a community-cured database on BGCs, which you can do chemistry prediction, and more recently, you can actually identify the boundaries of these genes, because before it wasn't so easy to know where one gene ended and where the other one started, or if gene clusters were part of a single pathway or not. And so from here, we are able to actually do annotate very carefully these biosynthetic gene clusters using a community-based standard annotation and metadata that is associated to BGCs and and their molecular products, specifically for bacteria. These two databases are available for everyone, and they're the basis for additional analysis downstream, which we are particularly interested in, which is network analysis through BigScape. And BigScape, this means biosynthetic gene similarity clustering and prospecting engine. And in a nutshell, what it does is it actually calculates distances between VGCs in order to generate networks that can then be interpreted in terms of neighbors of other VGCs that are nearby. For example, here we have a network that's a standalone network, GCF1, whereas GCF2 and GCF3 are linked by similarity. And this could mean that they are co-participating in a particular metabolic process or metabolic pathway. And from here, what we do is we use these networks and some of the elements of these networks to run them through Corazon, which is the third program that we use very often. And this was developed by Nelly Salem, a PhD student in our graduate program at Lancebio, which stands for Core Analysis of Synthetic Orthologs. And this program, what it does is it basically takes a query, one single gene that you're interested in, perhaps one of these elements from one of these networks, and it searches for homologs in all of these databases. And this would not be any different from traditional approaches, but in this case, what it also does is it generates a cluster variation database, which means that it actually takes this initial query and does multidirectional best hits to other genes that are in the vicinity of that initial query in synteny and flanking this particular query here shown in red, and filters by cluster homology. And so in this example here, what you see in red after the multidirectional best hits, you can end up with a list of genes that actually encompass a potential biosynthetic gene cluster. But the very unique thing about Corazon is that it doesn't just give you a list, which is many other programs actually did that before, but actually creates a phylogeny based on the concatenated genes that are in the vicinity of your initial query, which is what you see down here. And it also tells you how frequent these gene families or how frequent these genes and gene families are in your particular data set. So from here, you can imagine that you can, and this on the right is another query. This is the blue gene as an initial query. You can imagine that you can interpret these results, compare them by comparing them to the species phylogeny and to other phylogenies you might be interested in, and trying to see if these genes have a different evolutionary history than the species deeper history. You can also identify which changes have occurred and have this phylogenetic backbone that allows you to interpret any particular loss, gain, or mutation in these participating genes of the PGC. From here, you could also have the actual prediction of these metabolites and see if they might be part of a similar pathway. And this is also looping back to the BigScape and MyBig databases and platforms. So there's a suite of evolutionary genome mining of natural products, which is how we would generally refer as for these types of methods. There are essentially what we have very often we have a very clear biological hypothesis that we want to test by introducing an evolutionary element in these genome mining bioinformatics pipelines. So going back to CICAs and actually what we have found so far, the first thing, like I said before, we wanted to know if what was inside these correlate groups, because despite studies from even as early as 1875, basically, for at least 1 century in this correlate group, there was not much known about any bacteria associated to this group other than cyanobacteria. And even within cyanobacteria, it was not really well described in depth as we understand microbiomes today. So the first thing we did is look at their composition. And this is just one example of what we have found in the type of analysis that we do. These are metagenomic assembled genomes, or MAG. This is from a metagenomic sample. We assemble a genome using reference genomes usually, but also de novo, of the correlate root. And what it shows is a consortium of spectrum of genera, but some of them that are actually shared among all different samples. What you have here is different hosts, very different cycad genera from different places, and yet they share some of these bacteria that are the most abundant, such as Caulobacter, Nostoc, Calotrix, Pseudomonas, and usually many Rhizobiales, suggesting that indeed the main function of this root is nitrogen fixation, but it's not limited to that, and I'm going to get into that in a minute. We then look at these MAGs to search for biosynthetic gene clusters that could be unique to the group, to this particular cycad niche. And so here what you see is an analysis by Corazon, which I mentioned before, in which we constructed a phylogenate concatenating a series of proteins, or sorry, a series of genes that were unique to this niche, which are shown here in red and in black. This is unique to the cycads in general, and the red ones are unique to the species of Dionedule in particular, because we have several cycad hosts here. And basically what this allows you to find is these VGCs, which are here on the columns, that are only found in these particular samples and nowhere else in the planet, literally. So the comparison you see in this matrix is how can I find my biosynthetic clusters in these particular genes in other groups or other hosts compared to worldwide databases. And so this is a very useful approach when you want to narrow down candidates that you later want to validate in the lab. Now, I'm not going to go into detail on what we did to follow up the findings. You can read about it in our GBE paper in 2019. But essentially, this allows you to test hypotheses at the level of microscopy or mass spectrometry to actually look for the metabolites that are only found in this particular niche. We then wanted to know if maybe some of these BGCs are actually found depending on the type of soil or depending on the local environments that that cycads are living in. Because even though they all prefer calcareous soils, they actually do live in different types of local environments, including dunes, sand dunes along the beach versus oak forests in the mountains. So we compared four different sites in two different types of soil, clay soil and limestone soil, and we looked to see, to cut a very long story short, which we haven't published yet, what we found is we found different compositions of the BGCs depending on the soil type. We also characterized the soil, and the main element that differed between them was pH. So what we're doing now is we're trying to translate these genes into their chemical structure and actually try to interpret what the function of each of these gene components of the BGC could be doing for the cycad to be able to survive these particular soil types. But what I wanted to tell you more about today, and this is unpublished work that is very exciting, is that we also have been looking at the cycadivorous insects. And so these are all the insects that actually feed on cycads. And the very interesting biological question here is that how do these insects tolerate cycad toxicity? And the main hypothesis that we have is that it's able to sequester, inactivate, or transform all these cycad toxins. And so what we did is, in a collaboration with colleagues from Cornell and Harvard, what we did is we sampled insects across the world. This is work done mostly by Sheila Salzman and Melissa Whittaker, along with Naomi Pierce, to see if within the insect gut there actually could be a microbial or microbiome core that would tell us something about the role of these bacteria in detoxifying cytotoxicity for insects. And so what you see here is the worldwide sampling with different genera of cycads here shown in the phylogeny. And the different types of insects feed on different tissues of the plant, which you see here in colors. And this is just the structure of BMAA, which is the best well-known or the more well-known toxin in cycads. And so what we found is that there is indeed a very diverse microbial core, not as much as roots as expected. We know that in insects it's more limited in general, with several phylum that we knew, phyla that we knew were common, including in other insects. But what was really interesting is that we actually found a shared core of several bacterial genera among all of these insect samples of the world. And so what you see here on the left on figure A is just the general diversity of the various groups of these various phyla of bacteria in the different insect hosts. And what you see on the right is per insect, either butterfly, thrips, or beetle, what you see here is 4 genera that are shared in most, if not almost all of these samples. And that's what you see here in black. Where you see the gray is where it was found just once in the data gathered for that particular insect. And so in all, as the psychodiverse core bacteria is reduced to these Proteobacteria and Firmicutes, and these 4 that I have showed you here. So the natural question is, how conserved are their VGCs, and what about their function? Very similar to what we have done in the coralloid group. So again, we used the pipelines that I mentioned before, MyBig, Anti-Smash, MyBig, BigScape, and Corazon, to mine these metagenomes obtained from all of these samples. By the way, the information that I showed you previously is also it's a combination of 16net, amplicons, and metagenomic diversity. So what we found is that there are conserved VGCs of psychodiverse core microbiomes. And interestingly, they're highly enriched in siderophores, and particularly catechol types. These are usually antioxidants, so that's our main function found in plants in general. Hydroxymates and others that we call mixed ligands, carotenoid-like VGCs, arylopoline, and others that we are not sure what they are. So what you're seeing in this figure A is the different networks reconstructed by BSB. And another interesting feature of this is that most of these networks are isolated. And what does that mean? It means that the elements of the network, in this case, the various insects shown here under conditions, these are the insect hosts and colors, and the bacteria that is actually shown under organisms, are only linked among themselves and not linked to other potential networks outside of the cycad. So again, this is a comparison of the cycad samples and everything we recovered from the cycad samples compared to the entire database for secondary metabolites and predictions through BigScape and MyBig. And the fact that these are not connected to that other universe of possibilities means that they're probably very specific for bacteria associated with cycad. So the two very interesting findings here are that these are mostly siderophores and that they are mostly unique to this particular niche. And siderophores are interesting because they are basically small molecular iron chelators. And what does that mean? That just means that they can capture metals. And this is relevant because metals are toxic when they are not, when they're standalone, available throughout basically a cell. So these insects the toxicity for biotoxins in general and other elements in the cycad. So this is something that, of course, we still need to test in the lab, but this is a very nice prediction solely based on metagenomic data analysis and bioinformatic predictions. We went to look into Diuniduna more specifically. This is a Mexican cycad, and here we've essentially recovered a very similar taxonomic composition, which is what you see here. You see phylogenies of both butterflies on the left and butterflies on the right, on my right, and beetles on the left, weevils. And what you see here is the different abundances in the size of the circles and the phylogenetic distribution of this group. And what I just want to point out here is that there are the two of the core genera that were recovered in the previous analysis were also recovered here, which is Pantoea and Strenotrophomonas. The others were not recovered because this was a slightly different sampling approach. Here we did not go to experimental sample, so it's a little bit biased in that sense. But they are present, they're just not as abundant as in the other co-culture samples. And looking into the function of the most common BGCs within these samples from metagenomic assembled genomes, or MAGs, we found also that there's metal chelating siderophores involved somehow in these genomes, as shown by their BGC. So, in particular, within a bacteria called Pylobacterium, present in all the butterfly samples, there is always a siderophore that is a hydrosamate type, and it's essentially identical to the ones that we recovered from the previous analysis that I showed you. So, in all, this suggests that there is indeed a potential of a series of BGC adapted to this particular cyclic niche, both in the coralloid root. Remember, the first part was in the coralloid root, and the second part is in the insect gut. That is possibly playing an important function in the insects being able to be part of this biological system, which is the cycad, and the cycad being able to adapt to various local environments with different concentrations of metals. So the next step, we have a lot of next steps, including measuring metals directly from the soil and testing to see if you actually remove one of these genes, insects would then be vulnerable to these toxins, etc. So with that, I would like to summarize some of these results, and then I would be happy to discuss or answer questions. And now the take-home messages are that the rapid evolution of the cyclic microbiome and their metabolites, compared to the very long lives of cyclids, which can be, like I said at the beginning, 2,000 years or perhaps more, are what likely is allowing cyclids to adapt to very stressful local environments. And when I say stressful, I mostly mean low nutrients. But in the light of climate change, again, for cyclids, it could also mean other environmental variables. And they are likely the key to their long-term survival. So we have this holobionts with two rates of evolution, one under microbial components and the other one in the eukaryotes, that together actually make for a very resilient lineage. And so the very interesting aspect for me here is that the key endosymbionts are actually likely transferred and maintained in this cyclic system, from the soil to the plant to the insect and back. And this is important not just from an evolutionary standpoint, but actually it could allow us to understand other plant systems that we rely on. And I'm thinking in particular of agricultural environments in which bacteria are possibly being moved through insects into the plants, back to the soil, and so on. So this cyclic model, even though it's a unique, very particular lineage of plants, it actually can be a model to understand more open systems like agricultural systems, where we want to understand how plants are adapting to their local environments and to their insect pests, and what the microbial role could be in that adaptation. And on the other hand, environmental metagenomics, so sampling the records from the field, but supplemented with co-culture subcommunities, which is these semi-artificial environments that we have created or favored, along with some chemical prediction or chemistry, is actually a very powerful approach to understand the role of microbes in maintaining and generating biodiversity in general. But none of this actually would have made sense to us if we hadn't actually thought of biomimetic pipelines that allow you to detect these types of patterns in natural systems. Because as you might have inferred by now, this is a very long shot, or very far from other model systems in plants like Arabidopsis and other plants and other biological systems that are actually complex and difficult to interpret and difficult to test, to design experiments to test these hypotheses on. And so the bioinformatic pipeline then becomes a huge component of work. It's not trivial to decide how you will analyze this type of data, because as many of you may know already, when you're dealing with open environmental samples, you will get a lot of taxonomic diversity. And so therefore, pipelines that are guided through an evolutionary lens or evolutionary approach and that take advantage of the big data that's already out there and use it to parse an evolutionary signal are actually very powerful. And they're usually driven by biological hypotheses. So in this case, we had this idea that microbes are playing a very important role in these ancient plants that have survived essentially everything, except us in terms of their extinction rate. So that's the main message that I would want to leave this community, that when you think about pipelines, it's not just about good code, which is of course something you already know, but this is an example in which how you're analyzing and parsing data actually matters for the type of inference that you can have at the end of your study. And with that, I thank the organizers. I appreciate the invitation. I am not a bioinformatics, solely bioinformatics person, and therefore I am particularly grateful for this invitation. And here, this is just the funding agencies that have kindly supported this work. And with that, thank you.
Thank you, Angela. Very nice talk. We have time for a few questions, for some questions. And I would like to start. It's very interesting, the correlation between these metabolites in insects and plants. When you describe these BCGs, these clusters, what if you have a, for example, if you, some kind of the cluster is broken by genetic split away from different chromosomes, so the cluster is not complete. But if you get a cluster, you're not seeing the whole picture because there is a piece of the cluster somewhere else in genome. Does it interfere with this kind of analysis? That is, it is how it biases the analysis?
Yes, thank you. That is an important question. So basically, this would be correlation. Well, for the final interpretation, we would do that through correlation. I think that's the most direct evidence that we would have for what you're saying, which of course is something you expect, right? And what correlation does is it does a multiple hit, but it's not a bidirectional hit, but it's a multiple hit across all possibilities, and it goes from what you would assume is an ortholog, so a very high identity, to what normally turns out to be paralogs or genes that are already decaying. So you get the whole gradient of multiple hits. And with that, you actually capture things that have moved around. It doesn't matter where they are at the beginning, and it only matters in the next step where you actually reconstruct the phylogeny. And so the phylogeny, then, because you have to concatenate genes and they're ordered by similarity, you could actually see where something is missing. And in fact, you can actually see that things may have moved. So you see all the the horizontal transfer, because sometimes you see a gene present in a lineage, in a very, also present in a very distant lineage of your same database. And so you assume that was a jump or a horizontal transfer. And in terms of loss, you also see the empty spaces because you restrict it with a phylogenetic view. And this is why it's very interesting. This is why this tool is interesting, because otherwise, you're right. You would only get a list of only the positives, but you wouldn't get the false negatives. You wouldn't get the ones that are similar, but not similar enough to be able to get them. So yeah, you would miss all that information. But with this, you have the gradient of similarity, and then you constrain it in the phylogenetic structure. So you are going to be able to see, as long as it's sequenced. If it's not sequenced, of course, yeah, you won't see it. The other thing is, that's also why the networks, the Bigscape networks approach is interesting, because it actually does like neighborhoods of clusters. So you could, if you wanted, you could compare many replicates of the same thing, for example, individuals in a bacterial population or closely related population. and you could reconstruct the network, and then you would see elements missing because networks are closed in this particular method. Well, they're closed at some point, you know. I mean, if you expand the homology, then you can capture everything. But you will actually, for things that are meaningful connections, you will have a closed network. So you can see closed network 1, 2, 3, and then you see what's missing from one or the other. And then in that sense, you could also identify it there. You could also say, look, this is somewhere it decayed or got lost or it was deleted or it mutated into something else. Yeah. What's very interesting for me for the follow-up is now seeing the regulation of these clusters. Because if you think about it, we're not seeing anything about regulation. We just see presence, absence, but we don't know. Yeah, we don't know much about how they're regulated, and that's another maybe interesting tool that we can look into. I don't think anyone has developed it yet for bacteria. We do it a lot in eukaryotes, but we don't. I don't know if for bacteria. Yeah, I hope my answer was informative.
Yeah, no, that's good answer. Yeah. As can make another question. This is a striking observation that some insects carry the bacteria that is has a relation with the plants, and there is there is data about insects disappearing in some places and species of cicads or other plants that is also disappearing due to the extinction of this insect species.
Yeah, I don't have the numbers on top of my head, but these are all strict relationships. So that means if the plant goes, the insect in lineage goes, because they depend 100% on ficus. They're strict pollinators and strict herbivores. So they don't feed on anything else. They just feed on ficus. So yeah, it's immediate. If you lose a ficus, you lose a lineage of weevils or butterflies or thrips. Well, thrips are not as strict as beetles and butterflies, but weevils and butterflies definitely. Yeah. So you can just suppose that whenever ficus disappear, also their insect interactions, which is a shame because when you have a system with such a deep evolutionary history, you know, we're talking about, I said 100 million years, but most likely it's deeper. There's a bunch of really cool questions you can ask in this system, right? So, but yeah.
Yeah. Well, the cicads is in between us. It's around for so long time, and they probably had changed its patterns from it's insects, of course, but it's not the same species we have in millions of years. Just adjusting itself.
Yeah, it's the same lineage, but it's different species, of course. Yeah.
Yeah, the problem is the bottleneck. Sorry, sorry.
No, go ahead.
No, I mean the problem is the bottleneck we are posing to the environment. So now because if you have time and you have a healthy biome, a new species just replace other species, and the genera keep evolving. But if you just make a bottleneck, you may lose plants or insects, and then they're all forever. And
yeah, ficus used to be they're subtropical, so they're all in the subtropical range worldwide. And they used to be very widespread and very common when it was Pangea, when it was the one continent. And when Pangea divides, they're genera that are divergent and just stay in their own continent. They're very different from each other. But there's a huge bottleneck, like you said, at some point, several times in their history. And what we see now is just the result of a very recent diversification, which is like, well, it varies between 50 and 80 million years ago, with relatively new species and genera or subgenera that have appeared very recently. But that's in deeper time. But in recent time, in ecological terms, the populations that used to be hundreds of individuals for the most part, most of them now only consist of, I mean, there's a population in Mexico, for example, that is estimated in 25 reproducing individuals. So for the 20 plus species that we have in Mexico, only one has populations above 100, 150 that are continuous and healthy. So there's a second bottleneck, ecologically speaking, and with that comes all of the insects, which who knows what's happening to them. I don't think there's that much information, at least for Mexico, on that that I know of. Maybe there's some, but in general, I would assume that they're also going to be very strong bottlenecks.
Yeah, that's a concern. Do we have any other questions? Raquel, do you have questions? We have questions in.
I see some hellos, but I don't see questions.
Yeah, I don't then here.
All right.
Okay. Okay. I would like to thank you again, Angelica, for participating in the Brazilian Symposium by Informatics. I hope it's very interesting research. I hope you can get through this whole session, and I hope you have you back sometime. Maybe after this pandemic.
Yeah, maybe in person, right?
In a presential. Okay. Thank you very much.
My pleasure.
Okay. Thank you so.
Bye.
Okay.